Oh, hi. I didn't see you there. So welcome to my fourth video on cell culture techniques. My name is Erica Bowen. I'm a graduate student at the University of Georgia, and this is part of a series on how to do specific cell culture techniques in the lab. I have already made videos on how to aliquot, make media, and thaw cells, so today I'll be passaging cells. Currently growing out in my lab, I have a single T75 flask with really old D1 MSCs. I think they're at passage 17, so starting to show signs of senescence. Their morphology is changing. They're not wanting to adhere much. So I wouldn't want to freeze these cells back down and put them back in the cell bank to use for later. So at this point, I would trash them. And the reason why I was using such an old passage is for training purposes in my lab. Because I have those cells already growing out in a population going, I felt like it was a good opportunity to record myself passaging. So currently, they're not fully confluent in that flask either. They're probably at about 50% confluency. Passaging at anything below 80 to 90% is not really the most efficient way to grow out cells. Especially if you're growing out for a cell bank, you would want to let that flask reach 80 to 90% and then split into more vessels. Today, I'm just taking that one T75 flask and splitting it into two T75 flasks just for demonstration purposes. So if the T75 flask in that incubator is currently at around 50% confluency and I'm splitting into two new T75 flasks, then I will have a 25% seeding density.
Before beginning and showing the protocol, it might be valuable for me to run through some definitions again, and they might have come up in other videos, but I'll just go ahead and go through them again here if you need it. If not, you can skip it. So this first term I've already used just within the intro, and that's confluency. Confluency is the percentage of the culture surface area covered by our cells that are growing in adherence. It's used as a visual and functional indicator of cell viability, the growth state, and most importantly, your cell density. So it's something that we look at and sort of make an educated guess of how many cells might be present in the flask. So for example, in this image, I see cells here in my flask. These are D1 MSCs that I grew out in my lab. I would say they're not taking up a lot of surface area at all. I would call this 5% seeding density. Another person might look at this and think that that's 10%, and I would say that's valid too. This is a subjective practice. This image, I would say, is maybe 30%, and this one might be 80. These cells, I would call reaching confluency. So they're about at the point where you would want to passage them. When speaking about confluency, there are these terms, sparse and overconfluent. In a more generalized approach to cell biology, the cells in the ECM or the extracellular matrix talk to each other in a very specific manner. This is known as cell matrix interactions. They are also talking with one another via cell-to-cell interactions, and both of these are critical to maintaining cell phenotype and tissue function. So when you seed the cells too little or you passage them too early, you risk the cells dying because they're unable to communicate with each other and decide what needs to be done to live. Now, it is possible to actually seed from a single cell all the way to a full population. It's just difficult to do. This is called a clonal colony, if that's something that you're researching. But the cells don't like growing in that manner. They like having other cells to help signal to them like how they should be operating within a flask and within a culture. The opposite side of that coin is when the cells are overconfluent. So this is considered greater than 90%. Too many cells creates a competition of resources that can lead to cell death. So this is why we passage cells to prevent reaching this overconfluent status in which the growth rate is slowed, if not completely stopped because the cells have died. In this video, we are passaging. Another term for that is subculturing. It is the process of detaching and reseeding cells into new culture vessels with an S to prevent overconfluency and maintain healthy growth. This is an integer number that's representative of how many times the cells have been split. So in this video, I was working with passage 17 cells. That means they've been split 17 times, which is a lot for a cell line. Once I split them again in the video, they become passage 18. And really, this is used as an indicator of cell age and phenotypic stability. Once the cells start to become a higher passage number, you as a researcher can know, okay, these cells are probably going to start entering senescence soon, which is a decreased differentiation potential when it comes to stem cells, decreased ability to actually adhere to flasks and to surfaces, and of course, like a change in their physical appearance and function. This is important to track too, because if we want to compare data with other labs, we would want to know if you're using a passage number that's the same as our lab, because if you're using old cells, then they're probably going to behave differently than our cells, and we probably shouldn't be comparing our results one-to-one. My lab and a lot of labs use trypsin to detach cells from the surface. So this is the only reagent in this video that will be heating up besides media. And this is a cell dissociation solution. So when we add it to our culture, it will cleave cell to cell and cell matrix adhesion proteins in order to lift the cells off the surface and move them to a new place. If left on the cells for too long, just like DMSO, it is harmful for the cells. So this is why we have a process where we first lift those cells off the flask with trypsin, put all of that into a conical tube, spin it down, and then we can separate the cells from the trypsin and the media, where we can then remove the trypsin from the situation, resuspend in fresh media, and then move those cells into the new destination vessels. Also, in recording what I've done in the lab for passaging, there are certain points where my microphone has died. Yeah, I will be doing a voiceover for those parts, just as a warning. So here I was saying to go use the bathroom, come back in, and first step, put on PPE. So that's safety goggles, gloves, and a lab coat, long pants, and closed-toed shoes. Here I'm showing footage from the other day because I thought it might be more useful to show you what a proper flask looks like when you want to passage it. So these are like at 90% confluency. These are actually HMSCs, and these are very much packed in there and kind of forming the patterns that I would expect with those cells, so I'm comfortable passaging them. Then next, I'm talking about heating up trypsin, which is aliquoted in a 15 milliliter conical tube. And the only other thing you need to heat up is the media that you plan on growing the cells in. These reagents would take about 15 to 30 minutes to heat up. I'm turning on the water bath with the switch in the front. And while those heat up, I can go set up the biological safety cabinet. Just like previous videos, we're turning on the light, opening the sash to the lines on the side to establish proper laminar flow. I'm opening my biological hazardous trash and these biological sharps as well. It has reached laminar flow when the green light comes on. And now I'm ready to wipe down my working surface. So I'm spraying my hands with ethanol. And we keep an ethanol bottle inside of our biological safety cabinet. I'm adding the tube racks that I would need to hold my trypsin and a 50 milliliter conical tube. There's another tube rack in there to hold a microtube because I thought that I might also do cell counting in this video, but I think I'm going to make a separate video on that. I'm adding my pipette aid that was charging on the bench top. And I'm also adding serological pipettes in 5 milliliter, 10, and 25. I'm grabbing multiple in case something happens with the pipette and I need to get a new one. And then spraying those with ethanol and adding it to the biological safety cabinet. 50 milliliter conical tubes I already had open on our side storage, so I'm just going to add those to the hood. T75 flasks were not already open over there, so I will be getting a fresh bag from the wooden cabinets just across from the biological safety cabinet in our lab.
I'm also adding DPBS to the biological safety cabinet, and open ones are kept over here. Fresh ones are kept in the wooden cabinet. These are stable at room temperature, so it's okay to store them outside of the fridge and the freezer. And for all the open ones, you initial that you've opened it and on what date.
I also like to turn on the centrifuge at this point, and that switch is located on the back. And I'm going to counterbalance with 15 milliliters inside of a 50 milliliter conical tube because that's how much cell suspension I know I will have.
My media and my trypsin are completed heating up, so I'm going to wipe them off and bring them to the biological safety cabinet. So I took my bottle out of the water bath, placed it on a paper towel outside the water bath so that I can catch water bath water and prevent it from getting all over the counter. And I'm going to wipe it down lid first and then wiping away from the lid as to not be dragging contaminants up underneath this lip here. I'm going to spray this and add it to the biological safety cabinet. For conical tubes, it's the same kind of thing except holding it. Wiping down cap first. Being careful not to open this cap outside of the biological safety cabinet as I'm wiping it down. And then moving away from the lid. Everything is ready in the hood, so now I can bring my cells to the hood. I've already checked on them to make sure they were alive. When I reach into the incubator, I'm spraying my hands. And I'm handling the flask at the bottom, not touching this filter at all. I'm going to spray the flask and bring it to the biological safety cabinet. First step is to remove the media that's in this flask. If you don't have a vacuum, you will have to manually aspirate it. I have a vacuum. But with whatever you use, you want to open this lid first and work in these front two corners away from the cells that are growing on the back of your flask. I tilt the flask like this to bring the media into the working corner and then aspirate out that media. This tip never touched the cells, and I also never touched this glass pasture pipette to keep it sterile. The glass pasture pipettes that are kept in this biological safety cabinet have been autoclaved, so it's important not to touch it at the shaft or at the tip after that point, because then you're going to contaminate the pipette. Next step is to do a wash with the DPBS. I wash with half the working volume for a T75 flask, which is 5 milliliters. This step is important to remove any remaining media so that the trypsin, when added, actually works to detach the cells. If there's media or proteins left, the trypsin can be less effective and it could take longer to actually detach the cells than the 5 minutes described in the protocol. This is just a quick little editor note. You might see me throwing non-hazardous trash behind me. That's to get it out of the way. And when I'm completed, I will pick up all the non-hazardous trash and then go put it in the trash can that's by the sink. This is how I was trained in my lab. So I just aseptically placed a serological pipette onto the pipette aid. I'm not touching that at all. And I'm gonna cleanly pipette up 5 milliliters. And again, working in the corner away from the cells, place that 5 milliliters in the flask. This gets trashed. The inside of this flask, while not contaminated, is not clean. There are cells growing in here, so you don't want to use you don't want to take a serological pipette that has been in this flask and then put it back into your clean media or your clean DPBS. You might bring cells with that serological pipette. It's not good to get cells in your reagents. With the DPBS in there, I'm rocking it to cover where all the cells are. And now I'm gonna immediately aspirate that back out. I'm done with the DPBS, so I'm gonna recap it. That's the only time I will use the DPBS. Aspirate this out from the corner. And these glass pasture pipettes are going into the sharps, not the biohazardous trash. Now with our clean cells, I can add trypsin. Opening the conical tube first, reopening my flask again, and I add 5 milliliters of trypsin to a T75 flask. This is just enough trypsin to do the job, but not so much that it's an excessive amount, because of course the next step is to deactivate this trypsin added with 2 times fresh media. So if I add a lot of trypsin, I gotta add even more media, and it just becomes a lot of volume to spin down. So adding the trypsin into that corner again, trashing that serological pipette. I'm returning the lid to the flask and again rocking it. And now I'm gonna place this in the incubator for 5 minutes to allow the trypsin to do its job. You may need a time on your phone the 5 minutes. I have a timer here that I'm going to start. And during these 5 minutes, I like to prep for the next step. So I know once I've detached the cells, I'm gonna check it on the microscope, bring it back over here, and I'm gonna deactivate with 2 times media. So I just added 5 milliliters of trypsin. That means I'm going to add 10 milliliters of media for a total of 15 milliliters. When I'm adding that media, I'm washing the flask to make sure that I'm getting all the cells off that flask. And then I'm gonna transfer those contents into that 15 milliliter conical tube. During this time, I can open the conical tube. I used all of this trypsin stock, so I can bleach this and throw it away. Leaving the cap not all the way screwed on, so we're not pressurizing conical tubes in the biohazardous trash when it gets autoclaved. And I could also optionally go ahead and add media to these flasks, because when I spin down this conical tube that has trypsin, media, and cells, I'll resuspend in 5 milliliters, and then I'll split that 5 milliliters into the 2 flasks. If the working volume is 10 milliliters and I'm splitting 5 milliliters to go into each flask, this means that I need 7.5 milliliters in each flask of fresh media. So I've loosened all my lids, attached a 10 milliliter serological pipette. I'm pipetting up 7.5 to put into each one of these flasks. I can use the same pipette because at this point in time, these flasks are sterile. There's no cells in them yet. So pipetting up 7.5. Okay, so now I'm trashing this. I don't need it anymore. And because I'm not immediately working with the flask, I'm gonna go ahead and fully close the lids here. All right, my timer's going off. It's time to check to see if my cells actually detach. And the fact that it's slightly more orange now and a little cloudy is telling me cells probably did detach, but you still want to check it on the microscope to make sure. If cells were very much still attached, you could incubate it for 5 more minutes or a couple more minutes. If it was like this, but then like tiny spots maybe still attached, you can do a 2-finger tap method where you take the flask, hold it parallel to the ground, tap it with 2 fingers, just to kind of jostle and get the cells off of the surface. Recheck it to make sure that it worked. And then if that didn't, then I would incubate again in the incubator. But these are detached, so I'm gonna go ahead and do the next step, which is adding 2 times media to this, putting it in the conical tube, and then spinning it down. At this point, I don't want you to stress, but it is important that you start working and moving semi-quickly. The trypsin detaches the cells from the surface, but it also, if left on too long, can be bad for the cells as it eats away at unwanted proteins. And instead of adding in the corner at this point, you're washing the back of the flask. So it may be hard to see with condensation, but I am moving this pipette back and forth while ejecting the media onto the back of the flask. I pipette some more up, wash it again. I usually do this 3 times. Once I've done it 3 times, pipette all of it up. And then I'm moving it to the conical tube. I'm finished with that serological pipette, so I trash it. I'm bleaching this flask. Sometimes I keep it to remember what passage number it is, or like this information. But because I've already labeled these new flasks, I already know what is about to go in here. So I can just throw it away at this point. No longer need the rack that was holding the trypsin, so I can also simultaneously take that out as well. Okay, so now this is balanced against that 15 milliliter conical tube I put in earlier. This is closed all the way. And we want to run this 1200 RPM for 5 minutes. I wait and make sure that this isn't out of balance, and we're good. So now it's time for the last steps. I've taken the conical tube out of the centrifuge, put the balance back, turned it off. I've turned off the water bath because I'm done with that. And I forgot to hit record, but I aspirated out the old trypsin with media, left the cell pellet, and then I resuspended the cell pellet in 5 milliliters of media. I'll probably show an old video of me doing this already. And now I'm ready to split this into my flask. So I'm going to put 2.5 cell suspension in each flask, and that will bring each flask to the 10 milliliter working volume and split 50-50 among the flasks. So first, I'm gonna open my flasks. The conical tube is already open. I'm gonna mix it a little bit because it's been sitting for a second. And what I'm gonna do actually, and if you're comfortable doing this, is pipette up the entire 5 milliliters and then split it as I'm seeding it just with one pass. Here very carefully, putting this in the corner, and I'm pipetting down to the 2.5 mark very slowly. And the rest of it goes into this flask. There was a little bit of bubbles during that process. I didn't put it into the flask. I'm just gonna trash it. There are cells in those bubbles, but I don't wanna put the bubbles in the flask, and the cells that are lost in those bubbles is pretty negligible. And then as a last step, rock it so that the media containing the cells coats the entire surface area that you want cells to be seated on. And there's my cells. Now I'm gonna check to see if cells actually made it in this flask and if it looks pretty similar across the flasks. Seeing cells in this flask. Here's that in 4x. Yeah, there's cells here as well. One by one, I'm gonna spray these because they were individually sitting on that microscope, which is not clean. Spray them and put them in the incubator. Again, handling them by the bottom. Okay, now we're transitioning into cleanup. I've already turned off the water bath. I'm gonna close the centrifuge and turn it off. The microscope needs to be turned off and recovered to prevent dust from getting on its objective lens. I'm gonna start taking all my things out of the biological safety cabinet. Because I used the reservoir this time, I need to flush my vacuum. If you don't have a vacuum, you don't have to worry about this with ethanol. So it should still be on at this point. And pulling a little bit of ethanol in. Sometimes I like to plug this hole to kind of get it to travel all the way through in the Chameleon Lab vacuum. Recap this. This has been wiped down. I turn off the vacuum. Close the sash. UV light it because no one's going after me for further sterilization of the inside. And now I'm going to empty out the traps. Sometimes both need to be emptied out down here. We have 2 in the Chameleon Lab, but just this one seems to need emptying out. Want to turn on the water and do it a specific way. And I am allowing the water to run and help dilute this as it goes down the sink. And I leave the water running while I refill it with bleach located here. Enough to restore what was at the bottom. This is important to decontaminate any like aspirate you might have from your work. And I come over here and turn off the sink, and now I'm going to re-plug this back up. Make sure that you fully return the plug to this flask and connect this connector. Otherwise, it'll have a weak pull for the next person. And finally, pick up any trash off the floor if you operate like the Chameleon Lab does, to make sure that's clean, and recap the biohazard trash can so that you are compliant to the regulations of having a biohazardous trash can. Pick up these off the floor, and this can go in regular trash.
So that pretty much concludes passaging cells. Once these cells are growing out, you want to change the media every 3 days. You want to be checking the cells, honestly, every day to make sure that they're still living and nothing has happened and no contamination has occurred. if contamination has occurred, you should notify your PI or lab manager and bleach that flask and throw it away immediately. We change media every 3 days because media, or all the media that I've worked with when growing out cells is not stable at 37 degrees Celsius in the incubator like that. So you have to change it frequently, even though your cells might not be done using all the nutrients in there and in your media hasn't turned yellow. You still need to be changing at least every 3 days, sometimes every 2 days. So that's just something to note. And coming in to check every day too, to monitor its progression to confluency, especially if you're working with new cell lines. You're not gonna know for sure when they reach 80% confluency, and it would really suck if you came in and they were overconfluent all of a sudden. So thanks for watching.